Today, an increasing number of promising therapeutic approaches are in the pipeline, with potential to tackle the multimodal aspects of ALS and other neurologic diseases. What is the state of current and emerging pipeline? How are we addressing the complexity of biology? What about new targets and biomarkers? Please welcome Neil Cashman, CSO, Pro-MIS Neurosciences, Professor Emeritus, University of British Columbia; Michael McGrath, UCSF Emeritus Professor of Medicine, CSO and Founder, Navivo; Robert Scanneven, CSO, Verge Genomics; Fernando Vieira, CEO and CSO, ALS Therapy Development Institute; and Mulali Gopalakrishnan, Global Head, Neuroscience Search and Evaluation, Corporate Strategy Office, AbbVie.
I'll kick it off today. This is really an exciting time to be discussing on Therapeutics Pipeline in ALS, discussing actually and sharing the progress, particularly in a number of fronts that we'll cover during this webinar. First of all, there's been, over the past decade or so, increased understanding of disease biology and mechanisms in ALS. The field is coming up with very innovative and novel approaches for cellular and in vivo models for target discovery, validation, including utilization of some of the AI, ML type approaches to expedite early phase of drug discovery. There are pioneering new approaches for identifying the right patients and the right time with the use of biomarkers and translational strategies. And then there are a number of innovative therapeutic approaches that are in the pipeline that offers the potential to tackle the multimodal aspects of the disease. So these are a number of cardinal topics that we'll cover in this webinar today. We'll take a deeper look at all of these aspects in the coming minutes. So we're going to start off this session with Fernando, who's going to begin by outlining sort of the state of the emerging ALS therapeutics pipeline and some of the approaches that he and his team has taken recently. Fernando.
Thank you, Murali. And thank you, Wuxi, for the opportunity to be a part of the panel today. It's a pleasure to be here. Like all of my fellow panelists today, I've been in the ALS drug discovery and drug development space for a while, more than 20 years. And the clinical landscape has, in some ways, it hasn't changed enough, right? Ultimately, we still have a critical unmet need for people living with ALS, but in a lot of ways, it's evolved dramatically. The clinical development pipeline is much, much richer than ever before. There are simply just more companies and more drugs being developed clinically in different phases of development and preclinically. Our understanding of the disease has evolved very much as well. It's really, and when I say understanding of the disease, really understanding of ALS as multiple diseases in some ways. And I think that's developed far beyond what at least I could have imagined, 15, 20 years ago. I never would have pictured the landscape that we have today. And I think that development has come from, it's been rooted in a few important advancements. I guess a lot of it is a better understanding of diverse genetic causes of ALS, particularly in familial ALS, that 10 to 15% of folks who develop the disease who directly inherit it from somebody in their family, from their parent. So we've learned a lot from that. And we also have a better collection of tools and technologies that can be brought to bear to better learn about the disease, both at a basic research level and also at a translational research level. So now we have a lot of companies and even nonprofit organizations like ours at the ALS Therapy Development Institute that have clinical development programs with treatments that target a whole gamut of possible approaches. So yes, we can go after specific genes in the case of monogenic associated ALS, but we also have therapeutic approaches that go at ALS as though it's a proteinopathy, focused on maybe, aggregates of SOD1 or aggregates of Tdp-43 or the C9orf72 dipeptide repeat protein. So that's one approach that we're seeing a lot of. There are approaches targeting, metabolic function, mitochondrial function. We see a lot of therapeutics going after immune dysregulation, seeing if there are ways to modulate those pathways in ALS. And even more recently, we're starting to see targets going after gene splicing dysregulation. So it's a really diverse collection of biologies that have been implicated in ALS. And there's solid evidence to support a lot of these approaches. They may all have some value and some validity. And the key challenge might end up being finding the right people and the right time in which to intervene in all of these specific ways. It may turn out that not all people will respond to all therapeutic approaches. So you kind of have to think about the Venn diagram and overlap between the folks with familial ALS and specific mutations and folks with sporadic ALS that may have these different pathways engaged in these different commonalities, which I know a lot of folks here will talk about today. I can talk a little bit about how ALS TDI is going at this.
That'll be helpful. Yeah, that'll be very helpful, Fernando. Yes.
Thank you, Marley. So I guess for us, our approach is we're a nonprofit, right? So we're a nonprofit biotech or research institute. And our focus is to try to discover and invent or help other organizations advance their own technologies or drugs for ALS. And then partner in a way that gets those into the for-profit sector and develop them clinically. What we do, the way we do it, we have a research lab here in the Boston area where we use a diverse collection of model systems, cell-based systems using induced pluripotent stem cells, other cell lines, and animal models, including zebrafish and mice, to really try to discover new drugs and optimize them through preclinical pharmacology, and then get them to an IND-enabled drug. And we're modality agnostic. So we have one example of a drug, a monoclonal antibody targeting CD40 ligand. So this is an immune target, which we've been able to partner and move into clinical development. Phase 2A trial just read out. It was good. It was promising. So that's one way that we've gone at it. But actually, our preclinical pipeline right now is focused on small molecule drugs, targeting mitochondrial energetics or post-translational modification type called protein arginine methyltransferases. So we think it's going to take a lot of different approaches, diverse modalities, diverse targets, to really ultimately address the needs of everybody living with ALS. And we're really excited about the idea of working with others to make that happen.
Thanks, Fernando. That's a very, very nice overview. And you did put the overarching aspects in perspective. So thank you very much for that. Just to continue on the same theme, and Fernando touched on the importance of targets and going after the right targets, mentioned about the genetically defined targets, in this case, the familial disease. Of course, the sporadic population is significant. And many of us know that the biology underlying Tdp-43 is pretty fundamental to the overall pathophysiology of what's going on. So I want to maybe turn to Neil now to sort of elaborate a bit more on what's our understanding on Tdp-43 biology. How are we tackling that aspect of the biology, either directly or indirectly, and share your perspective on, what are the challenges in this space as we explore the biology around Tdp-43, which is a very fundamental component of the disease pathology. Neil.
Thanks, Murali. Tdp-43 has been implicated in many diseases, especially ALS, but also frontotemporal dementia and some other neurodegenerative diseases like the late phenotype of Alzheimer's disease. Tdp-43 is, we all understand what you and Fernando are saying, that there is quite a bit of heterogeneity in ALS, but we're not confined to treating each and every individual variant of ALS, because there are some features of all or virtually all ALS that are preserved. And one of those is Tdp-43 aggregates in the cytoplasm. And this is interesting because Tdp-43 also can have familial ALS mutations that lead to the disease, but the vast majority of Tdp-43 aggregates are wild type protein that have not been mutated. So it may be kind of on the razor's edge for being involved in all or most cases of ALS. So the pathophysiology of Tdp-43 basically has two facets. One is loss of function, because in ALS and in frontotemporal dementia, there is nuclear depletion of Tdp-43. This eliminates Tdp-43 from performing its normal functions, particularly important for neurons, which include RNA splicing. We might hear a bit later with Rob that Stathmin-2 and UNC13A are some of those proteins that have been implicated in ALS, which undergo splicing changes with the depletion of Tdp-43, nuclear Tdp-43 in disease. But there's also other losses of function. Tdp-43, normal Tdp-43, is engaged in RNA transport, stabilization, and even protein translation in distant compartments like the synapse or the dendrite. You can't knock down Tdp-43 to avoid the cytoplasmic aggregates because of these vital, critical neuronal activities that it supports. Mm-hmm. So what are the gains of function? These are basically marked by cytoplasmic aggregation, which appears to induce inflammation via at least two pathways. And we'll hear from Mike about the role of inflammation in disease, but also promotes mitochondrial dysfunction, ER stress, proteasomal stress, translational stress with ribosomal collisions, the whole gamut. Tdp-43 aggregates also have a surface that recruits co-aggregation of nuclear transport and pore proteins, alpha-synuclein, stress granule proteins, but it's a poorly recognized gain of function that may also produce the loss of function of depletion of nuclear Tdp-43, and that's via its prion-like activity to recruit new copies of Tdp-43 to the misfolded aggregate. So how do we tackle Tdp-43, this pathology, the loss and gain of function? Well, directly, if we had a method or a drug that would break up aggregates and repatriate Tdp-43 to the nucleus, that would be great. Mm-hmm. So that is a pathway that we're following in PROMISE neurosciences to selectively target misfolded Tdp-43 with antibodies and intrabodies. Indirectly, one can eliminate the negative consequences of loss of function of Tdp-43. And we'll hear from Rob at Verge about iPSC technology to replace or allow Stathmin-2 to be expressed to stabilize axons, and UNC13A to promote vesicle genesis in the nucleus. Robert will also talk about a kinase inhibitor, which is very important in the pathology of this loss of function.
Yep, it's very helpful, very, very helpful. Maybe we could close with, what are the challenges in this space with Tdp-43? Certainly an exciting target, but with respect to the challenges in targeting Tdp-43, can you maybe make a closing comment on that?
Sure. So the indirect approaches are the most advanced, but it's unknown if correction of one pathway will lead to an amelioration of disease, although it is very hopeful. Mm-hmm. For our specific targeting of Tdp-43, the challenges may include vectorization of interbodies, which will be a gene therapeutic, which has really never been tried before.
Yep.
So these are the hurdles as I see them.
Great, thank you very much. And so Neil touched very much on sort of the role of the neuroimmune mechanisms in the overall pathophysiology. It's not unique to ALS, but I'd like to invite Mike McGrath to sort of elaborate more on the role of neuroimmune mechanisms in ALS, examples of targets and approaches, and what are the most advanced efforts in that space. Mike?
Yes. So many years ago, I started looking at neurologic disease, neuroinflammatory disease, by looking at AIDS dementia. There's a subset of AIDS dementia patients who had sort of classic ALS kind of symptoms. When the effective therapy for AIDS came along, I said, I'm going to work on a nice, easy disease. And so I decided that ALS was going to be it, because it had a nice readout of the ALS functional rating scale. And I thought it'd be a piece of cake. Well, it turns out it's not. This is 20 years later. And but we have learned a lot. I think that so inflammation is a normal. It's normal. I mean, we're experiencing it every microsecond. You've got something, a bacteria gets eaten, it causes inflammation, and it recycles back, and it turns off the activation signal. And so the problem that we're having is that in ALS and a lot of these chronic diseases, that somehow this inflammatory signal is not getting turned off.
Right.
And so the question would be, are there different classes of signal that are not being turned off or regulated? And I'm, I did cancer work. I have a lot of my friends. I'm in the hematology and oncology division, and if you think about cancer, there's no one drug for cancer. In fact, people will like throw stuff at me if I said I have a drug for cancer. So I think that we've learned from the sort of the cancer environment and are moving it towards now the ALS environment, and saying that there are factors, whether they be genetic factors, mutations that are passed on in families. I don't know any little children that get born with an SOD1 mutation that have ALS immediately. And so this whole notion about the time wherein something makes you predisposed to a disease, what is going on that leads up to the actual manifestation? So we've really taken a step towards saying, we're going to look at the most inflammatory people and learn what's causing those inflammatory changes. And from our experience, I think that almost anything can incite inflammation, whether it be aggregation of proteins, the genetic abnormalities that one sees, misfolded proteins, damage. I mean, soccer players, who knew you get 8 times the frequency of ALS in football players with trauma. So you have these little micro leaks that lead to central nervous system problems. So anyway, I think the approach that we've taken is trying to understand chronic inflammation. And one of the drivers of inflammation chronically in the neurodegenerative disease is microbial translocation of the gut, where you have low levels of endotoxin in the blood in all these diseases, whether it be Parkinson's, Alzheimer's, ALS. And that's a process that keeps this inflammatory loop going. And so we and probably others have been trying to figure out how to put this under better control. And I think that I'm excited. I think we've got a path forward looking at regulation of trying to remove this chronic drive of inflammation. But anyway, so we appreciate that there are different inciters of inflammation, but we're going after what the common features are in inflammation. And I would say that I don't know what the proportion of patients are where inflammation is actually the major driver. I think you have to start somewhere though. So I think that just as a final point, the FDA guidelines came down in 2019 where they acknowledged that ALS is a heterogeneous disease. And if you can take a big group of patients and then focus in on a subset and say, these are people who respond to this approach, and I'm going to design a trial and fix that group. And that's taking a personalized medicine. That's exactly why almost 80% of all the new drugs that are being developed right now are cancer drugs, because that's exactly what people do.
Right.
And so that's, I'm going to leave it there for a time being. I'd love to hear what Rob's working on as far as his the genes and regulation that they're looking at.
Yeah, that's very helpful, Mike. Well, maybe one question before we go to Rob. When you think about neuroimmune mechanisms and the therapeutic approaches, do you see that obviously coexisting with other therapeutic targets and approaches as well? So would it be sort of a foundational approach with respect to the neuroimmune targets and the biology, and then of course there are other factors impinging on that? Is that the way you look at it?
Yeah. So I had the toughest talk I ever gave in my entire life was my daughter's 5th grade class, and I said the brain is like a city, and the houses are the neurons, and the way they talk to each other is the little roads and sidewalks between, and the brain's important because it has these houses. Now we think that there's a fire that's going on and blocking the ability for people to walk between the houses. And if you want to apply BDNF, growth factors, things like that, you have to stop the fire. And so I think that the approach is very simplistic, of course. Stop the fire using something like our drug, our approach, or any of the anti-inflammatory approaches, and then try to rebuild, because we know that nerves regrow slowly, but they can regrow. So the point is, let's stop something and then build on top of that. So it's not a single answer. It's going to end up being cocktail for sure.
Great. Very, very helpful. Very helpful. Let's go to Rob, actually. And speaking of sort of the next generation targets and the biology, Rob and his team is taking some very innovative approaches with respect to AI/ML, with respect to target validation. So welcome, Rob, and I'd like to hear your perspectives on the next generation of therapeutic approaches in the ALS space. Rob.
Great. Thank you, Murali. I think as we heard from many of our speakers today, ALS is a complex disease. We know that it arises from an interplay of genetic drivers, some of which are known, many of which are still probably unknown, and the interaction of these drivers with a host of other genetic and environmental factors. Looking at individual genes and individual points of therapeutic intervention in isolation may not be providing us with a complete picture of the multitude of pathological processes that are occurring simultaneously within patient cells. An alternative mechanism for discovering new therapeutic targets utilizes an integrative multi-omics approach in which we capture multiple different domains of biological information, such as genetics, transcriptomics, proteomics, and metabolomics, which can improve identification and characterization of pathological processes within disease-relevant cells. Deconvoluting these complex and very large omics data sets requires advanced AI and machine learning techniques to really parse them and plumb them for their deepest meaning. So for example, at Verge Genomics, we've developed a computational engine that can decipher complex omics data and identifies dysregulated networks of genes and proteins that are enriched in disease patients as compared to healthy controls. We can then perform deep bioinformatic analyses on these dysregulated networks, which promotes discovery of new therapeutic targets. And these targets are predicted to broadly impact and reverse multiple pathological processes within this dysregulated network. So beyond the identification of new therapeutic targets, integrative multiomics is also perfectly poised for new biomarker discovery, as proteins or metabolites that are dysregulated in disease can be candidate markers that allow for evaluating the effects of new therapeutic interventions on targets, so target engagement markers, looking for markers, the effects on disease-relevant pathways, and markers of disease processes such as neurodegeneration. So the same AI machine learning techniques that can identify new targets within dysregulated networks are also equally adept at identifying new biomarkers. So as an example of this process, at Verge, we've used this approach to identify a previously unknown pathological process in ALS. So analysis of perturbed gene networks in ALS patients identified a particular enrichment of genes involved in endolysosomal function that were centered around C9orf72 as a genetic driver. Subsequent studies at Verge and follow-on work from multiple other labs has confirmed that C9orf protein, which as Neil mentioned, is downregulated in patients carrying a C9orf72 repeat expansion and represents a loss of function phenomenon. This work has characterized C9orf72 as having a critical role in regulating intracellular membrane vesicle trafficking in endosomes and lysosomes, which impacts multiple different processes in the cell, such as autophagy. At Verge, we identified a therapeutic target which was derived from this dysregulated endolysosomal network. This target is PIK5, which is a phosphoinositide kinase. Inhibiting PIK5 activity can broadly reverse endolysosomal dysfunction, and we have a lead candidate that's advancing towards the clinic. We validated PIK5 as a target using multiple different systems, including human patient-derived systems, such as ALS patient iPSC-derived motor neurons. And we also use mouse pharmacology models of ALS, many of which Fernando's organization works with extensively. We and others are demonstrating that the integrative multiomics approach allows us to decipher complex biological pathways and overlapping pathological mechanism. This then aids in the identification of new therapeutic targets, which we believe will have a greater chance of translating into the clinic and produce disease-modifying effects.
Well, it's very, very helpful, and like the way you laid out, really capturing the disease biology into identifying not only compelling targets, but also biomarkers, which kind of goes back to where Fernando started. How do you really sort out overlapping mechanisms and approaches for combination, and coming back to identifying the right patient with the right therapeutic? So it's a step in the right direction, but I want to maybe pause here and have, I don't know if Fernando wants to come back and comment in terms of how do we piece it all together, because right now we have the technology, the way of approaching it that's coming together, along with the aspiration to do some of those studies and put it into practice. Fernando?
Yeah, I think the ALS space is in a much better position than it was years ago, in large part because of a number of sort of collaborative consortia, sort of pre-competitive consortia, a lot in the nonprofit space that have played a pretty big role in collecting large data sets that give opportunities for lots of players in the space to really hone in on patient subsets and try for biomarkers for patient stratification approaches. And so we at ALS TDI, we have a program which we call the Precision Medicine Program, which is rooted in that. It's 8 years running, collecting, it's like a natural history study collected with whole genome sequencing, connected to induced pluripotent stem cell biology, connected to longitudinal blood sampling for biomarker discovery. But we're not the only ones in the space. There's, Target ALS has done similar types of things, another nonprofit. Answer ALS, largely associated with Hopkins, and many other clinics in the United States have done that. I know Canada has recently initiated a similar type of program, and I think we see something similar in Europe as well. And I think more data sharing, more bringing these types of information to the fore for industry and academic researchers will help us get across that finish line in terms of finding the right patients for the right drugs and the right targets.
Yeah, absolutely, absolutely. And following up on that, as those studies get underway, obviously one of the continued questions we wrestle with is endpoints, functional readouts. How precisely can we measure the role of digital approaches? Maybe someone would like to sort of elaborate perhaps a bit more. I don't know whether Michael would like to jump in and sort of elaborate on sort of the landscape with respect to functional endpoints and clinical readouts. Michael?
Yeah. So I think that it's pretty critical to figure out you've identified a subset of ALS patients and you have a drug that you think does something. I think that a part of what's missing is proof of target engagement. Like, are you actually hitting what you think you're hitting? And that's the strength of TDI. I mean, no question about it. I mean, you guys have screened a ton of different things and you have a very set series of criteria to say, this is actually happening. And it's a good thing. So one of the things that we've done most recently is looking at very objective measurements of function, in this case, respiratory function, so that you can actually map a single nerve, which would be the phrenic nerve, more or less, looking at respirations using a spirometer, and get hard endpoints that way. So to the extent that you can get a hard endpoint tied with biomarkers, I think that you're really going to move the field forward. And so that's the other thing that Rob's over there. No, whatever, which direction Rob's in. Okay. He's developing his omics approach. I think that that's a very powerful approach. And if you are working with groups that have, and you probably have got your own drugs, just set the bar high about saying, I have to have proof of target engagement using my multi-omics approach before you move to the next step. Because I think that's going to be really a much faster approach to identifying effective therapies.
Great, thanks. Rob, would you like to add anything further from your perspective?
Yeah, I think to expand on what Michael was saying, the deployment of new digital biomarkers, I think, really would be transformational for the way we think about clinical trial endpoints. The example that Michael spoke about of spirometry, which has been, a gold standard in the ALS field. There's some really recent work that suggests that you can get similar type of data with an app on your phone looking at sustained phonation. And it tracks very well with spirometry. And this is looking at this voice analysis can do something at home that you would otherwise have to go into the clinic or work with a skilled technician to accomplish. So I think it's these endpoints that allow us to look more frequently and interact with the patients more frequently, collect more data, really will transform how we think about trial endpoints in the coming years.
Very helpful. Thank you.
Yeah.
Neil, any comments from you to add, Neil?
Just as somebody who's a clinician scientist, at this point, we've come a long way in trial design and in measures of response. I forget who it was who mentioned, but the ALS-FRS, which used to be regarded as a neither linear nor ordinate, has kind of proven its chaps at this point to be responsive to therapeutics. But it really is the advent of biomarkers. I haven't heard one mentioned yet, neurofilament light, NFL, which also appears to be treatment responsive in motor neuron diseases. So we're at the point where we have a good scale, finally, a good clinical scale, finally, and we have an existing biomarker which can give us the target engagement or involvement in biology of disease of these various therapeutics. Of course, the future is coming fast, including the digital biomarkers, but at this point, we have these two useful measures of response.
Thanks, Neil. It's great. So for the remaining time, I'm going to assume that I'm an ALS patient, and I'm going to ask the question, what can I expect in the next 5 years? What is your vision for how the pipeline or the therapeutic pipeline is progressing? And with a view from the patient, what can I expect for a viable treatment? What are the mechanisms that you like, for example, or which are the ones that are progressed further along? It will be helpful to get some guidance and insights from each and every one of you. So I'm going to maybe start with Fernando, who is actually on my left. Maybe start from Fernando, and then we'll go through each one of you. Fernando.
Sure. I think at the top level, we can hope and expect a few new approvals that are already in the pipeline. There are a few things that are under FDA review right now, which have shown some promise clinically. And we can hope for new options to be available for people right now. We don't expect that those options are going to be transformative, right? I think what's in the pipeline right now there are some therapeutic approaches that have potential for profound effect on some people. And there are some therapeutic approaches that have potential for a good effect on many. But we don't have many therapeutic approaches that have potential for a profound effect on all. And so I hope that that we can strive toward sort of that clinical toolkit that can get eventually to profound effects for all. But it might be a complex cocktail, and it might be different for one patient to another. So I think an ALS patient might expect for more sampling to happen in them, right? So over time, we may end up seeing more genetics play a role in their diagnosis and prognosis, and then therapeutic targeting, and maybe assessment of biomarkers like neurofilament that Neil alluded to, seeing if you can get a prognosis on disease progression, and then bending that prognostic curve for each of those people with drugs in the clinic. So it's a better time than ever, but not good enough.
Great. Well said, Fernando. Maybe pose the same question to maybe Rob.
All right. Maybe take a slightly different approach. And I think one of the more exciting developments in the past few years is the emergence of platform trials and other large research groups and initiatives that are really going to allow us to centralize a lot of the information that we receive from trials. The Healy Platform Trial Group in Europe, we have NCALs and TriCALs. I think these groups are going to be transformative to how we think about data collection in our trials. And they also represent an advantage for patients in that the structure of at least the Healy platform is that you have a significantly greater chance of going on to a therapy versus being in a placebo group, and the ability to enroll in multiple trials. And I think that's going to represent a great advance for patients.
Thanks, Rob. Maybe pose the same question to Neil, and then we'll go to Mike. Neil?
Sure. So I agree with my colleagues, but I heard Fernando use the word hope. And this is what I convey to my patients. I've been in the field for almost 40 years, and there's never been a time in history when more people are working on ALS and more companies are in the space for the purpose of developing and testing drugs. So this is, as Fernando said, there are 2 compounds right now under consideration at the FDA. That's also been something that I've never experienced. So there's a profusion of new approaches. There's an incredible amount of activity and funding. I think that the prognosis for the next 5 years is very good. So I think we're in for something good in ALS.
It's very hopeful. Thank you. Mike, do you want the last comments here on this topic?
First of all, I think that it's incumbent upon us to figure out how best to use ALS patients, very valuable but short period of time, where they can actually be in a clinical trial. And that's why I think that moving towards more of a little bit like a cancer approach and say, all right, what's an acceptable response rate, whatever that response is, and define it with some kind of an objective measurement. In cancer, you look at, did it shrink or not? Well, we're taking a look at this whole spirometer kind of a thing, just saying we're looking at one lower motor neuron endpoint, and that is the functional respiratory function. That plus biomarkers tracking along together, because I think that it's important that you don't do a clinical trial where somebody occupies 6 months to a year of their life. And I think that Rob's comment about the Healy platform, I think the platform is spectacular. Great idea. I would also like to encourage there to be other sort of unique approaches, the kinds of things that Fernando's been doing, but have short-term biomarkers as a readout and tell the patients to say, you know what, you're participating in early steps of something, and we need you for a month or something like that, because we want to try to validate a target. It won't keep you from being in other trials. But I think that with this new way of looking at digital biomarkers, things like that, we've got to accelerate the endpoint readouts so as to encourage pushing some of these things through across the finish line.
Great. Well, these have been very, very insightful comments today. We heard about how emerging biology is being tapped into with new approaches, the multiomics, the AI/ML approaches. We heard about commitment from not only companies but also foundations and other organizations. The need for increased collaboration in this space. So overall, this has been a very illuminating webinar. I really appreciate and thank the panelists for joining today for this conversation. And I hope those of you who are watching it do really enjoy this discussion as well. With that, we'll bring this to a close. Thank you.
Thank you, Murali.
Thank you.